Hello, everyone. First of all, thank you very much, Natalie and James, for the invitation to and for organizing the whole seminar series. It's very nice, and it's a very nice opportunity to share our work. So the focus of my talk today is specifically on the use of human lung organoids for the respiratory disease modeling. In terms of a very quick introduction, advanced non-malignant lung diseases are life-limiting respiratory conditions with limited number of effective treatment available for the patients, high morbidity and mortality, low number of available lung transplants for the qualifying conditions, and poorly understood pathogenesis stimulate great interest in generating new in vitro models to study the human lung. A variety of platforms have been developed, including two-dimensional and three-dimensional culture systems to derive specific cell type and structures of the lung. To further our understanding of human lung development, disease, and regeneration. So the idea here is that a direct differentiation of human pluripotent stem cells, which are unspecialized cells that can give rise to particular cell lineage, are considered to be a highly reliable system complementary to animal models. As a result of human pluripotent stem cells differentiation toward lung cells, we can create human lung organoids, which are self-assembly structures that accurately recapitulate the histological and functional aspects of the in vitro tissue and can, of course, provide a source of lung progenitors that have a potential for cell therapy and a platform for disease modeling, which is actually a case in our lab. We are referring here to pulmonary fibrosis. That is the topic of the presentation. So what are exactly those pluripotent stem cells that are used to generate human organoids? Those are the cells which came from the inner cell mass of the blastocyst during human development. And those cells have a great capacity to cell renew by dividing to develop into the three primary germ layers of early embryo, endoderm, mesoderm, and ectoderm. And therefore they can differentiate into all cell types of the adult body, but not extra embryonic tissue as the placenta, for example. So additionally, induced pluripotent stem cells are a type of pluripotent stem cells that can be generated directly from somatic cells when we put additional reprogramming factors. They have both type of pluripotent stem cells. They have a very high potential as experimental model for research purposes. And in the case of our lab, we are studying human lung development and disease modeling. So a mammalian respiratory system is extremely a complex structure which consists of a tree-like arrangement of branch airway tubes which are connected to a single trachea and terminating in millions of delicate, highly vascularized gas exchange units, which are called alveoli. The epithelium lining the whole system is continuous and differs actually significantly along the proximal distal axis, both in the cellular composition and structural organization. Coordinating the development and homeostasis of an organ as complex as the lung requires critical cell intrinsic and cell-cell interactions. To achieve those complexity, a mammalian lung undergoes a progressive development process, starting with the specification of the endoderm progenitors in the anterior foregut, which is followed by the postnatal maturation of the gas exchange interface. Both processes are further defined by specific cellular tissue development events, such as proximal distal patterning of the endoderm and mesenchymal, to specify large and small airways, branching morphogenesis, and the airway and postnatal alveolar genesis. In this context, our lab followed the differentiation pattern of human lung development And we established an experimental protocol from human pluripotent stem cells, which allows a direct differentiation into progenitors. To go more into details, let's look at this slide. Here we started with a two-dimensional lineup, which was then adapted to a three-dimensional system, leading to specification of the lung field, which allows to grow in suspension so-called lung-bud organoids. Once those lung-bud organoids, LBOs, are embedded in the matrigel, they can undergo branching morphogenesis and model human fetal lung development. In terms of characterization, LBOs are characterized by the presence of pulmonary lung field and endoderm marked by NKX2.1 and FOXA1 respectively, and some mesodermal component that is represented by PDGFR alpha in this system by immunofluorescence staining. Further, if we are embedding the organoids in the matrigel, they display branching morphogenesis over time with up to 77 days just featured over here. And further, when characterized by the immunofluorescence staining, they display distal maturation marked by the surfactant specific markers such as surfactant protein C, B, or ABCA3. So in this context, if we developed a nice model of human lung development, we wanted to go further and apply this system for respiratory disease modeling. In this case, idiopathic pulmonary fibrosis, IPF. So what is this disease exactly? This is a progressive and ultimately fatal disease that is characterized by fibrotic obliteration of lung alveoli, leading to respiratory failure. Unfortunately, therapeutic options are extremely limited, and the only definitive treatment that is available for now for those patients is a lung transplantation. So far, the etiology and pathogenesis of IPF are still unclear. Here we can see the most commonly known risk factors. The risk factors are age, environment, and genetics factors. Some studies also highlighted that IPF can be caused by repetitive alveolar epithelial cell injury and dysregulated repair. Here we can have some potential targets for the therapy and prevention, either the epithelial compartment or the proliferation of mesenchymal cells that lead to deposition of collagen and further IPF development. The most important part is that type 2 alveolar type epithelial cells are potentially highly involved. Those evidences came from the subset of patients with Hermansky-Pudlak Syndrome, HPS, which is a similar clinical entity with IPF and is caused by abnormal abiogenesis and trafficking of lysosome-related organelles. HPS patients are characterized by pigmentation and bleeding abnormalities that are associated with dysfunction of melanosomes and platelet granules, respectively. In the same line, those are similar to the lamellar bodies of alveolar type 2 cells, where surfactant is stored and recycled. The mutations that are causing HPS affect 4 distinct protein complexes, and out of 10 that are known recently, only HPS1, 4, and 2 are associated with features of pulmonary fibrosis, which is called Hermansky-Pudlak Syndrome interstitial pneumonia, HPS-IP. So, in fact, that only some of those HPS mutations are associated with pulmonary fibrosis, offer us a unique opportunity to determine features of epithelial cells that are shared by those with mutations that predispose to HPS-IP. The idea of the experiments were that we introduce IPF-associated and non-associated mutation in embryonic stem cell lines, using CRISPR-Cas9 gene editing technology and characterize hPS mutant lung organoids and with transcriptomic and phenotypic approaches. Moving to the results, here we can see the bright field images of day 40 material embedded lung organoid culture that were generated either from Y-type parental line or hPS mutant lines. And those data demonstrated that hPS 1, 4, and 2, they showed abnormal compacted morphogenesis, morphology, sorry, for when we compare with hPS 8 and Y-type lines. We wanted further to characterize the protein expression in those models, the immunofluorescence staining from mesenchymal markers in Y-type and mutant organoids. Those data revealed increased deposition of collagen 1, 3, and fibronectin, also an increased presence of cells expressing mesenchymal markers such as Vimentir or PGF-beta alpha, and finally the mesenchymal transcription factors such as SLAC in hPS 1, 4, and 2, but not hPS 8, that is not fibrotic-prone line, compared to the also compared to the Y-type. We selected some markers that were confirmed with Western blot. And then those observations were also quantified from the immunofluorescence staining over here in each mutated clone, and further with RT-qPCR. So in this context, we again can see that hPS 1, 2, and 4 mutant lines, when we compare to the hPS 8, there was no differences compared to the Y-type, which is actually in accordance with the clinical profile of hPS-IP. We further selected another approach that can help us characterize the protein profile in this system. So you can see the representative flow cytometry plots of the 40 Y-type and hPS mutated lung organoids that were stained for epithelial and CD90 mesenchymal markers. And here again, an increased CD90 population was found in fibrotic-prone organoids, hPS 1, 4, and 2, compared to the hPS 8 and Y-type. In addition, we compared the genome-wide expression in epithelial cells from day 40 Y-type and hPS mutant organoids with the key gene database, and we found that we matched the results with the second trimester human lung. Here, we decided to complete our data with the RNA-seq analysis. Since the pathogenesis of IPF and hPS-IP involves epithelial compartment and epithelial injury, we perform RNA-seq on the purified epithelial cells from organoids that were generated from Y-type and from each clones to really identify gene expression pattern that differs from parental line and are shared by the fibrotic organoids. And here you can see that there's 56 total genes that were found to be significantly upregulated in both hPS 1 and 2 that are the fibrotic-prone lines, but not hPS 8 compared to the Y-type, while 28 genes were significantly downregulated. Among upregulated genes, interleukin 11 was of particular interest for us as it is a soluble cytokine. So we wanted to investigate more in details what is the effect of the cytokine and how it responds to how it behaves in our model. We characterize interleukin 11 expression. First of all, we found an increased expression of the cytokine in fibrotic-prone conditions by RT-qPCR in the transcriptomic level. Then we quantify the protein level of interleukin 11 in the supernatant of hPS1 organoid culture by ELISA. And finally, we saw an increased expression of interleukin 11 in IPF and hPSCs patient samples quantified by in situ hybridization. In addition, we wanted to examine whether interleukin 11 can drive fibrosis in our model. We culture wild-type organoids in the presence or absence of interleukin 11. And we can find that the organoids that were treated with interleukin 11 shared increased expression of mesenchymal markers by immunofluorescence and quantify on the left panel and quantify in the middle panel, confirmed by RT-qPCR. Then follow up, we decided to delete the interleukin 11 in hPS4 knockout fibrotic-prone organoids that displayed actually morphology that is comparable to the Y-type model we have in our hands. So double knockout organoids revealed the disappearance of the fibrotic markers that we quantify all over the clones by immunofluorescence, and here on the right side again by RT-qPCR. So all those data confirm for us a central role of interleukin 11 in the pulmonary fibrosis. To summarize our model of lung-bud organoids that we have in hand with our protocol, we can easily create a lung-bud branching morphogenesis and that are enriched with the mesenchymal cells. We don't need any reporter lines or forced expression of transcription factor to evaluate the effect of the factors of interest. This works only in the matrigel, does not work in other matrix. For this type of organoids, we can successfully model the disease, and we don't need any serum in our culture system. That is a very good sign for the potential clinical applications. However, we have some limitations. This is the only lung cell organoid culture, so there's no other vasculature or other different kind of cells like immune cells. There's no terminal maturation of the system, and we have a bias toward the distal differentiation and epithelial type 2 cells. The developmental stage is still fetal. It's not an adult model of lung organoids. So here, in conclusion, for my talk, we actually modeled successfully the pulmonary fibrosis in lung organoids that were generated from embryonic stem cells with mutation in hPS genes that strongly predisposed to this disease. And the second point that is important is we demonstrated an essential role for interleukin 11 in the fibrotic process. What is interesting in this, the second point, is that it aligns well with the recent data that were published by several other groups in terms of effect of interleukin 11 in implication of the cytokine in pulmonary, cardiovascular, and liver fibrosis. So we really think it's a potential target for fibrotic conditions for the patients. Here, with that, I would like to finish and acknowledge all the members of my lab, especially my advisor Hans Snook and Alex Strikolis, who actually perform most of the experimental work for this project. I would like to acknowledge our collaborators and founding institutions. So thank you very much for your attention, and I'm happy to take some questions.
Thank you very much, Anna. That was a really interesting talk. So for anybody that has any questions, if you want to post them in the chat function, I will read those out for Anna. So I think James has some questions he wants to eagerly ask, so I'll pass you over to him for now.
Well, you did actually answer quite a lot of my questions. It was a really nice presentation. I was just curious, you mentioned at the beginning about the environmental factors. And whether and how this could be used to test environmental factors on the effect of environmental factors on fibrosis. Are you doing this, or have you had any thoughts about it?
So actually, we are not investigating environmental factors. We just take them into consideration when we try to plan our experiments. So one of the factor that could be really affecting the progress of the disease is smoking or any other asbestos effect. So whether if we have some clinical data, we will pay attention to the smokers, no smokers, and how the disease is developing. But by our hand, we are not really investigating in this model this kind of risk factors.
Sure, and I mean, just as a follow-up, I guess, if people were to investigate environmental factors such as smoking, is there any move towards making like a sort of open organoid structure? Because I guess at the moment you'd be delivering environmental factors to the outside rather than to the inside of the lung. Do you see what I'm saying here?
I think I have an idea what you try to ask, but and I try to figure out how potentially it could be developed in that way. So we have other projects that are related with the organoids, and if we put any other factors not actually related to the fibrosis, we mostly affect the media. So we will just dissolve the factor in the media and just evaluate what is the effect of the expression of the proteins or on any other pattern that we are interested in. I'm not sure if we can directly inject something like that in the organoids. The thing that worked in our hands to inject was the viral infection, but any other factors I'm not aware that we were trying or we are actually thinking of implying. So
that's fine. Sorry to take a tangent there. I was just interested.
Sure.
And then you mentioned about the limitations of there's no immune cells, no vasculature. Again, in other organoid fields, people are looking at co-culturing these kind of systems and building these elements into their organoid models. Is there any work towards that end?
Yes, actually, we have two postdocs in our group. It's Ivana, who is developing vasculature system that will be co-cultured with our organoids. And we have Remy, who is developing the macrophage lineage that is going to interact with the lung organoids. But so far, we don't have any data to present for that. But this is something that the work is in progress, and potentially there will be something happening soon.
Yeah, I think that could be really interesting. And then I was going to ask about the matrigel, about whether you can work with synthetic materials instead of matrigel. But you mentioned at the end there it doesn't work with collagen or lung ECM.
So it depends which kind of pattern we are interested in obtaining. The pattern that we show for the disease modeling is working the best in the matrigel. We had also some results with collagen one, but then the pattern of the lung organoids actually change and it gets much more differentiated towards the distal pattern. So a lot of alveolar type 2 cells, much more than we see in this. So if we change the matrix, actually we can change a lot of the expression around our organoids. It affects a lot. So yes, it's possible for other systems that we are working, but for this disease that we were trying, matrigel is the best working option for now.
And I was going to say, is that sort of still under investigation, or is that known what factors in matrigel are effective in promoting this effective structure?
We were discussing, but we were not really going to details for that. I cannot tell you the composition of matrigel that could really affect our system, because it's also probably a mix of matrix or a mix of cytokines in the media. So I cannot give you the details, but probably there's something that is just in the composition that works better in this case.
Okay. Can I ask one more, Natalie?
Sure, go ahead.
And then with you sort of mentioned that some factors push it sort of more distal. I guess, are you looking to sort of take inspiration from developmental biology and introduce different factors? And I guess that's been part of this whole process along, but is there a good understanding about which factors push the organoids in different developmental directions?
So what we are using is actually the mix of cytokines, and now we have another postdoc who is evaluating more in details the cytokine composition and the patterning and the lineage development of the organoids whenever we remove or we keep the cytokines of interest. So as what I just briefly can mention, that FGF7 or FGF10 affects the patterning of the distal differentiation. Also, the share that is the agonist of the GSK pathway that is really affecting, and we had a model that is working well in the collagen. When we remove the share from the media, we have extremely high differentiation toward distal lung development. So,
all right, yeah, yeah. Thank you for answering the questions.
Thank you for questions.
Back over to Natalie.
Thank you, Anna. So we have one more minute. So some of the kind of questions I had have already been answered and quite related, but I was also interested in, based on the change in the ECM deposition, is there a way of like measuring how that might be differentially affecting the mechanical properties of the environment around the organoid?
So this is a very big challenge for us actually, because the fibrotic organoids, they already secrete some mesenchyme around, and it's more enriched in the mesenchyme markers. But material that we are actually using for embedding, it's also very rich on a lot of markers that interfere with our organoids. So it's extremely technically difficult for us to quantify more precisely how it behaves. So the idea for us now to try to quantify these interactions by light sheet microscopy. So just see through the organoids how the markers are disposed and how it can change over the particular condition. But it's extremely technically, as far as I know for now, to determine the effect of the mesenchyme transition for those organoids.
Thank you very much. Yeah, I knew there'd probably be some technical issues with this. We have the similar problems in our lab as well. Okay, so I'm going to pass back to James to introduce Caroline, but thank you very much, Anna, for your talk. And if anybody has any other questions, I'm sure Anna will be happy to answer them by email.